Adhesion-driven vesicle translocation through membrane-covered pores. Picture a microscopic world where vesicles, nature's cellular couriers, traverse narrow pores to translocate across the plasma membranes of cells. Vesicles are bags made of lipid bilayer membranes that can enclose cargo to be transported between and within biological cells. Adhesion-driven vesicle pore translocation is not merely about pushing a vesicle through a pore. It's about an interplay of energy costs for vesicle and host membrane deformations and an energy gain for vesicle-host membrane adhesion. Initially, spherical vesicles need to deform considerably when squeezing through a circular pore with a radius smaller than the vesicle radius. In vivo, a pore that a vesicle translocating the plasma membrane of a biological cell needs to squeeze through might originate from a cortical cytoskeleton below the membrane. The dynamic voyage of a vesicle through a pore can also be used to understand better the importance of parasite deformation during the invasion into host cells. Plasmodium, which causes malaria, and Toxoplasma, which causes toxoplasmosis, deform considerably when invading their hosts. Interestingly, recent micrographs of Plasmodium merozoites that invade red blood cells show that most of the microtubules that stabilize the parasite shape are depolymerized, which strengthens the applicability of our vesicle model. Here, the pore is the tight junction where the parasites are observed to squeeze through at the host plasma membranes. The elongated shapes of the parasites might speed up pore translocation. Our numerical calculations allow us to predict vesicle translocation states, such as a free vesicle before translocation, a weak translocated vesicle before the majority of the vesicle has translocated through the pore, a deep translocated vesicle, and a complete translocated vesicle. We show that a finite host membrane tension hinders deep and complete translocation, requiring higher adhesion strengths to achieve these states compared with tensionless host membranes. The mechanism of host membrane tension reducing the invasion of Plasmodium parasites into red blood cells has been proposed earlier based on an experimental study. Vesicles are abundant in vivo and used as synthetic drug delivery vectors. The vesicles in our study also serve as a generic model system for elastic particles. Although it lacks various building blocks of real parasites, which we aim to look at in future studies, it can help us to characterize the effect of parasite deformability on the host cell invasion of apicomplexan parasites through the tight junction. Check out our paper for further results and details.